Okay, let's just dive right in. We are living through what a lot of people are calling a new psychedelic renaissance. Seriously, after decades of being in the shadows, things like psilocybin, the active compound in magic mushrooms, are totally back in the spotlight, and they're sparking this intense curiosity from everyone: scientists, therapists, and just the general public. And hey, this is way more than just some cultural trend. Right now, there are these super rigorous clinical trials looking into psilocybin-assisted therapy for a whole bunch of conditions. We're talking everything from severe depression to end-of-life anxiety. And the early results? They've been so promising that they're really making us rethink how we treat mental health. But here's the big question we're going to tackle in this explainer. The term magic mushrooms sounds so simple, right? But the reality is anything but. Are we actually dealing with one single, uniform substance? Or is that name really a doorway to a bunch of different worlds? To find out, we're going to zoom out, level by level, starting in the lab, then moving to the clinic, and finally to the home. So let's start at the most fundamental level, the mushroom itself. What is actually inside these fungi? This is where the story starts to get really, really interesting. A recent, and I mean incredibly comprehensive study didn't just look at one or two types of mushrooms. Nope. Researchers analyzed the complete chemical profiles of 42 different strains of these psilocybin-producing fungi just to see what was really going on under the cap. To do this, they looked at each strain's metabolome. Now, a metabolome is basically a unique chemical fingerprint for any living thing. It's a really cool concept. So just like no two human fingerprints are exactly alike, the complete set of chemicals in one mushroom strain can be wildly different from another. And this slide just perfectly illustrates how different they can be. I mean, look at this comparison from the study. On one side, you have Psilocybe cubensis, which was just packed with psilocybin. But on the other side, Paniolus sinensis had the highest levels of psilocin, which is the active psychedelic compound. Plus, this whole cocktail of other molecules that we haven't even identified yet. They're not the same thing at all. So the huge takeaway from the lab is this. The researchers concluded that we just can't treat all these fungi as one cohesive group. Each strain is its own little chemical factory, producing a totally unique blend of compounds that could have very, very different effects on a person. So the term magic mushroom, it's a massive oversimplification. Okay, let's move to our next level now and see how this all connects. We're zooming out from the chemistry in the lab to the therapy that's happening in the clinic, where real people are using these compounds to try and heal. This brings us to a Canadian study that looked at patients with cancer diagnoses who were given legal, compassionate access to psilocybin therapy. And this chart, wow, it shows just how significant the results were. Only 2 weeks after a single session, participants reported these major reductions in their anxiety and depression. But here's where that real-world complexity kicks in. The experience wasn't some silver bullet for every single person. While most people found it incredibly meaningful, the study is very careful to point out that one participant actually reported a decrease in their well-being. It's such a critical reminder that these are powerful experiences and they come with a whole range of outcomes. And that really highlights the difference between two very distinct worlds of therapy. In a clinical trial, everything is super tightly controlled: the patients, the dose, the environment, everything. But in the real world, like in that compassionate access program, you've got a much wider variety of people, conditions, and support structures. The researchers make the point that we just can't assume that the amazing results from trials will automatically translate to these less controlled settings. Okay, let's zoom out one last time. We're going from the clinic to the culture at large. And this is where we find this grassroots movement of microdosing. People taking these tiny, subperceptual doses for self-therapy, for enhancement, and they're doing it completely outside of any medical supervision. Research into this world shows that becoming a microdoser isn't some casual thing. It is a deliberate, multi-step journey. People start with awareness. Then they do tons of research and what sociologists call stigma work, basically actively reframing the risks for themselves. Then they have to navigate this legally gray world to get a supply, experiment to find their personal sweet spot, and finally, figure out how to integrate it all into their lives. So what motivates someone to go on this journey? This quote from a participant, it is incredibly powerful. For a lot of people, it's not a choice made lightly. It's coming from a place of desperation, where the potential benefits of trying an illegal substance just seem to outweigh the very real risks. The choice for this person was legally dead or illegally alive. So, the crucial takeaway here is that microdosing is not passive consumption. It takes a huge amount of active work and personal investment. These users have to become their own researchers, their own suppliers, and their own therapists, navigating stigma, finding a source, and meticulously tracking their own progress to try and separate the immediate effects from the lasting benefits. Okay, so we've journeyed through three really distinct worlds: the chemical world of the lab, the therapeutic world of the clinic, and the cultural world of the home. Let's try and bring it all together. This table just brilliantly summarizes our whole journey. In the lab, we learned that magic mushrooms aren't a monolith, they're a diverse collection of unique chemical fingerprints. In the clinic, we saw that while therapy can be life-changing, real-world outcomes are complex and they're not guaranteed. And in the home, we discovered that microdosing is this active, user-driven process of self-experimentation and a whole lot of work. And all of that leaves us with one final massive question. These three worlds, the precise science, the supervised therapy, and the grassroots user movement, they're all starting to collide. As they converge and start shaping policy and medicine and culture, the challenge for all of us is to figure out, how are we going to navigate this new psychedelic landscape wisely?